Hi, this is Dr. Joe Chow speaking. Today, we would like to introduce a new instrument called PAMPA dissolution, uh, especially for the generic drug bioequivalence study and 505(b)(2) drugs R&D. Here is my vita about my experience in pharmaceutical research for first in Sandoz pharmaceutical company in the United States, and also I have like about a couple of years in Pfizer as a senior scientist for the new drug development. So about 60 of the generic drug was successful filing while I was working in Sandoz. Okay, and here in this case, before the PAMPA was introduced, I would like to talk about something regarding the IVC, so-called in vitro to in vivo correlation mass model. Therefore, that will lead to the design of the whole instrument. As this PPT shows, you can see that the generic drug bioequivalence is actually consists of two parts. One is so-called in vitro, one is in vivo part. And we all know that the in vitro part, as the pharmacopoeia indicated, right? So we had to meet that in vitro requirement for drug release. However, prior to that, you need to meet the in vivo, means bioequivalence study. But how to arrive that is the topic really very important as far as today, because by tradition, we are always using pilot bio study. That skill is a very traditional skill, may not really help people out. It give us a scientist like a rough direction in terms of estimation of test drug versus reference drug. However, in this PPT, we would like to show the more scientific way in terms of predicting the bioequivalence of test versus reference, so that this equation is being like developed in terms of like IVC wise. So as you can see, the very bottom, the drug absorbed F equal CP times PE times area. As you can see from these upper color squares, the F, the drug absorbed, is equal to the drug concentration in the GI tract times the intestinal permeability of the drug times intestine surface area. As long as we have these, equation like clear, then we know that there are parameters to be monitored, which is CB, meaning the drug concentration in the GI tract, and the PE, which is the permeation of the drug in the GI tract. And since this equation is really validated through several publications, back in 1990. Okay, so we will go through these later. Now we will talk about something dissolution and drug absorption in terms of if you have a tablet or capsule that is being done through this pilot bio study. In terms of the tablet capsule, as a subject, you will need to take 240 ml of water, and that drug will dissolve in the stomach and finally absorb in small intestine. That is usually the process starting from tablet capsule through all the way to intestine, right? But can we really do that in terms of equation-wise, so that at least we know how to monitor that, right? In terms of like drug absorption-wise. Okay, so therefore we need to develop the model. This model was developed back like 1990 by certain like scientists in literature. However, here we want to show how this thing was done so that the audience has a clear idea about how important is this model. So initially, as you can see from this figure, that tubing like figure mimic this small intestine. It has a length of L and has a drug mass rate in from left side, Q times C0, and out is Q times Cn. And there's some drug, it will be permeate to the side of the small intestine. So we can write such in and out and permeation mass balance, which is equation 1. Like you can see, the drug absorbed F equal dN divided by dt equal to Q times C0, which is left side, minus Q times Cn, which is right side. So this purely is just a mass balance, meaning like the drug coming in from left side of intestine and coming out from right side of intestine. And their difference is whatever lost, meaning whatever absorbed. So that can be done in terms of mass is JW times dA and double integration. So that we can do integration easily, right? It will become like equation 2. So as you can see, after double integration, F equals JW times A. However, you can see JW is a wall flux, A is absorption area. So then, we need to solve this JW. And fortunately, we found that JW equal CB times PE, where CB is the drug concentration in the GI tract. However, PE is the permeation of the drug. So we substitute that 3 into equation 2, give us drug absorption, which is F equal CB times PE times A, which is A is a surface area of small intestine. So by using such an equation, we will know that we need to control the parameter of CB and PE in small intestine, so that we know we can arrive with the drug absorbed as compared to the reference, so that this derivation-wise, we can see from the next PPT to further prove this equation is valid. Okay, this PPT shows further derivation by substituting this equation 3 to equation 1 and expand the surface area. So you will have equation 4, and that equation 4, after this substitution, and we will further divide it by Q and then divided by C0. Now it becomes equation 5. So equation 5, basically you can see clearly that the drug absorbed, meaning F equals such expression. And as you can see from here, the Q can be further substituted, and this CB becomes CB star and DZ becomes DZ star. These are the star meaning dimensionless. That was really just pure mass transform through the division by C0 and this L. So it becomes like meaning dimensionless for integration part, yet there is a variable, it's called PE, and these are all constant. And after substituting this Q, we can see from next transparency about how this becomes. Now the equation becomes like this. Substituting Q with this parameter into equation 5 give us equation 6. And this equation 6 can be, further symbolized by using an equal such a group here. So equation 6 then becomes like equation 8. So it's just, a simple transformation, easy, becomes like F equal to an integration of such, right? So we know that integration of this is purely just a constant. Now, F is a variable depend on 2an, which is like depends on PE, because others are all constants. So from the equation A, we know that they are only like a simple algebraic relations between the drug absorption and PE, so that we can draw a line easily, right, by this equation A. So that simply because this is just like high school math, this like looks like y equal to x times constant. So we know that there is a simple relationship between y and x, and you can draw a line through, right? So the equation A is validated then via the market drug in the United States. So that means the whole derivation of the IVSC model is really validated. You can refer to this article. There are fully description as how this thing was done, right? So then we know such a model can be further derived in terms of using the machine-wise, right? Because such a model is really controlled by this equation, meaning there are two variables actually. The equation A was like for the validation purpose, but in the reality, what we need to use is we need to use a machine which can monitor the PE change and versus the CB change. That means a concentration change and the permeation change in one machine, so that we can really monitor the two variable, variations during these dissolution. So there's such a machine was further developed using what? Using first the bioequivalent media to control the CB, not the buffer solutions. It must be using bioequivalent media, because the bioequivalent media really mimic the GI media. Okay, the buffers just for the purpose of FDA reviewing, and really it's just a tool of QC release. Okay, so by using such a bioequivalent media, then scientists in the laboratory can easily understand how that CB was really affected, right? And in terms of P-wise, we can use a so-called PAMPA test. That PAMPA test, it was developed back like maybe 30 years ago. Until today, we combine these two parameters together so that the way of predict the BE can be feasible by using first bioequivalent media and the second PAMPA test. Okay, so which means the skill also meet the IVIC model. So interestingly, like we can see the PAMPA dissolution therefore was invented, in terms of B study prediction. Okay, so let's see what's going on about PAMPA. The PAMPA really look like this, as a figure indicated. You have a dissolution media in the yellowish part of the graph, and there's a filter impregnated with so-called phospholipid solution, and that used as so-called PAMPA membrane, and your solution components, meaning your API, will pass through this unstirred water layer through these membrane wise, and then pass through the UWL again, being absorbed, okay, into these plasma. So that if we are using such a configuration, then we can easily, using so-called PAMPA dissolution, to predict your drug absorption in the GI tract. Actually, the whole graph here is try to mimic the real situation happen in the GI tract. Okay, so this so-called unstirred water layer actually is so-called unstirred. Yes, if it is unstirred, the thickness usually is about 1500 nanometer. Okay, however, in the real situation, in testing, it's about 300 to 100 nanometer, that range. Therefore, you need to stir to reduce the thickness of unstirred thickness. Okay, and that applies to the both sides of the UWL layer. Okay, all right. With this fully understand of this PAMPA theory, and that was studied back 30 years ago until today. So it was fully studied by scientists. There are numerous literatures that we can refer to. You can or just Google PAMPA, this word. You can find lots of studies in this area. Okay, so now till now we understand all these this IVIVC model, and we have understand all these PAMPA principle. So we come up with this so-called the PAMPA dissolution figure. Okay, so as everybody understand that in the dissolution, let's say apparatus 1 or 2, we have 6 vessels on the top, and then we have a pump in the middle, and the PAMPA underneath. Okay, so with respect to each of these vessels, it has each of respective PAMPA. Okay, so through pump, the solution was pump like back and forth, just to meet the requirement of dissolution and requirement of permeation. And therefore, with time going, we can measure the change of the drug dissolved and also the drug permeated. Okay, so that CB and PE can be like measured with respect to these fiber optics, so that once you drop your tablet or capsule into this apparatus, dissolution apparatus, and drug will be slowly dissolved and being pumped to the PAMPA, and the drug will penetrate through the membrane measured in the pink area, which is the plasma, and being measured through the fiber optics, and so as to this CB. So we have these fiber optics installed. Okay, so by using this graph, then we can easily measure the change of CB and versus PE, right? And that will be meeting the requirement of this model, which is drug absorbed F equals CB times PE times A. So that CB and PE now is really under the machine control. and it can indicate clearly what's going on about the reference drug, and therefore your test drug, can be really compared to the reference. So in this configuration, really what we do is that for the, let's say, dissolution vessel 1, 2, 3, we put the reference drug, and 4, 5, 6, we can put the test drug, so that we can compare simultaneously using one machine and so-called PAMPA dissolution to complete the comparison between the CV and PE parameter with respect to reference drug and test drug. Okay, so we can see now the outcome of case 1 study, which is Mycotis 40 milligram tablet. Generic name is called telmisartan 40 milligram tablet. As you can see, the left graph is the CV. That means this drug dissolution. And the right graph is PE. That means permeation. So as you can see, we have 1, 2, 3 is channel is Mycotis. Yet in this case, we put identical 4, 5, 6, the same lot of Mycotis and same strengths. As you can see, their dissolution curve almost identical and their permeation almost identical. Okay, that will just prove that the machine design was correct. Okay, if the machine design isn't correct, then therefore, therefore you cannot predict the drug difference, right? So in this case, we can see clearly that the dissolution and the permeation, they are all the same with respect to the same lot of drug. Okay, so next, transparency for the ezetimibe 10 milligram tablet for the PAMPA dissolution. As you can see, same graph for CV and PE. Here, the red line is the reference drug, and the blue line is Sandoz drug. And the outcome is, let's see, from this permeation, you can see the red line is the reference azethol and the Sandoz drug, and they're fairly slope-wise, right? If you do the regression line of all the points, you will see the slope-wise, they are fairly close. Slope meaning this, the y-axis divided by the time in the x-axis. So y divided by x meaning the slope. So that means that the unit time, how much drug was absorbed. Okay, so that means if your slope comparison of the blue to red, then you can end up with prediction of the Cmax, right? Because that's unit time of drug absorbed. So you can compare the slope of the blue versus red, you will get your Cmax comparison easily in terms of predicting the human absorption Cmax. However, for the AUC wise, how you're going to do that is really simple as the red line AUC. That means the curve, the area under the red curve. So you can do like a triangular integration of the red and versus triangular integration versus blue, right? So we can compare the area. So in this case, you can see that, definitely just by eye, you can see their slope, they are fairly close in terms of Sandoz versus azethol. So you can see that this Cmax, it will be passing. And the AUC, by integration, we know that the Sandoz will be slightly lower than the red one, right? But after integration, we found that it will be above 80% also. So we know that in this case of comparison, from the PAMPA dissolution, we know these sandals will meet the requirement of both Cmax and AUC. So by using such a simple machine that will exclude the human variation, because human does have a subject variation, and we want to avoid that. Okay, so we can use such a machine, the so-called PAMPA dissolution, to help us in the prediction of bioequivalence in the laboratory. Okay, so in this case, we will end up with like a table for us to predict the Cmax and AUC, and we can see if you have a drug, let's say Tmax of 1 through 5 hours, usually, for the IR drug, immediate release drug, they are all like, we need to have a guidance, able to predict the Cmax and AUC, right? So such a table was generated, and then in terms of predicting the Cmax, we can use slope ratio, as we discussed, and the AUC ratio, definitely we can use that for the prediction of AUC requirement. And then the difference is such, and there are reasons behind. But definitely, I mean, if you can adjust your formulation to overlapping these two curves, then you will definitely meet the requirement, right? So that's what is very convenient for scientists in the laboratory using such a dissolution, PAMPA dissolution. You will, as soon as you have the graph, then you can, do next day formulation adjustment and do the test again. So that will save your time and money. You know what I mean? And in this case, let's say Sandoz versus Azathrol, we can see that it's fairly easy comparison and quick comparison. On the same day of experiment, you will have the data already because this is fiber optics measurement. Okay, so this is a very quick and very scientific way in helping the scientists design their formulation. Okay, all right, so as far as to clear the difference between the PAMPA and the human PE values, here we point out like 6 cases of drugs. As you can see from this particular drug, they have different, strains and definitely different BCS class, right? And like these SCT hydrochlorothiazide case-wise, we have this measurement value via PAMPA, and this is the clinical literature data. So as you can see, they are very close respect to each other, same as the rest of different drugs. So that give us a proof that the PAMPA system is quite dependable because it's been really studied in the scientists by the scientists throughout maybe 30 years. So it's a mature technology. However, to combine both together respect to this IIFC equation, such an instrument is just developed within like past 2 years only. Okay. As far as this PPT says, is the B prediction method. By tradition, that's left side, and by PAMPA, that's the right side. What this PPT show us is this. By tradition, if you do the pilot bio study, usually we have, need to recruit about 10 subjects, and the whole time of length is about 30 to 60 days, And, through this, even you done the bioequivalence study, right? And still that will have, the data variation because of the formulation variation plus subject variation. They are all together. There are two variations, that will cause this BE data, right? So then if you are a scientist to read this BE data, it combines both variation. So how would you be able to tell? It is because of the formulation variation, or is because of the subject variation? That leaves scientists still a question mark. That's why by using this traditional prediction, your accuracy is lower than 50% because you have two variations combined in the B data you receive. So you're still need to guess what's going on, right? However, if you use PAMPA, then there's no subject variation, it's only formulation variation. Then that gives us an easy reformulation because you have data direct, P comparison, CV comparison, and PE comparison shown, right? So you can easily compare these two formulation difference, and you can rely on the data and make change of the formulation easily. And that's why it says here, the PAMPA excludes subject variation effect. And therefore, you can do your formulation easily in your laboratory. That will save lots of time and reduce your risk, right? So your passing rate will be a lot higher. Okay. Let's read the conclusion of the the prediction methods. Traditional method, pilot bio. Yes, I mean it helps, but medium. In cost wise, is the highest, and length wise is longest. But if you are using IBIVC prediction, yes, I mean nowadays you can do it like in 2 weeks, less cost. However, the prediction accuracy and reformulation, they are low because of these in vitro dissolution may not be accurate. Okay. However, with PAMPA dissolution now, we know we have this validated equation, and based on the validated equation, we can have these two parameters measured exactly and without the influence of individual subject variation, right? So therefore, you have data, and therefore your prediction is high, and your reformulation is also high. So that means you can easily do your reformulation, at laboratory. And these PAMPA data can also be used in Bio5B2 reformulation design also, not only the generic drug, okay? So with all these indicated, as I said, the PAMPA method becomes a feasible and very dependable method in today's formulation development. So that's it. I will stop this introduction of PAMPA dissolution for today's introduction of the PAMPA for everybody to get familiar with this new instrumentation. And I hope, if you have any question, you can email me. And hopefully one day this instrumentation can help everybody in terms of designing your formulation. All right, thank you. And you have a nice one.